All right. Good afternoon, everyone. We're going to get started in just a moment, but before we do, a few logistical details. We are recording the event, and the video will be available to view from the Basic Science website in a couple of weeks. You can view this video from this event, as well as videos of the other events in the series this season and prior seasons. Due to the large number of attendees, we are going to keep the audience muted throughout the event, but we do want to hear from you. So if you have questions for any of our speakers, we hope you'll post them in the chat, and we will try to get to as many of them as we can this hour. And without further ado, it's my pleasure to introduce the Dean of Mathematical and Physical Sciences, Steve Kahn.
Good evening, everybody, and welcome to another episode of Basic Science Lights the Way. As Susan said, I'm Steve Kahn. I'm actually speaking to you from New York this evening, but I'm very happy to be here for our last event of the fall to share more fascinating stories of curiosity and discovery with you. Every great discovery starts with a curious mind. As Albert Einstein said, the important thing is not to stop questioning. Curiosity has its own reason for existing. One cannot help but be in awe when one contemplates the mysteries of eternity, of life, of the marvelous structure of reality. It is enough if one tries to comprehend only a little of this mystery every day. And that quote is at the heart of our Basic Science Lights the Way series. It's amazing where curiosity can take you in science, and sometimes those are some of the most interesting and rewarding directions. The theme of this series, as our regulars know, is basic science research, also called pure or fundamental research. We strive to show why this type of research is so important and why it is at the heart of what lights the way at Berkeley. Through open-ended research, we can refine scientific theories and better understand natural phenomena. Tonight's topic is invisible science. You'll hear about how some of our scientists perform science from the microscopic level to the Planck length, which is the smallest possible unit in physics. Once we're underway, I hope you will post your questions for any of our speakers in the chat box, and we will answer as many of them as we can. Our moderator this evening is Professor Russell Vance. He's a professor in the Department of Molecular and Cell Biology, and also a Howard Hughes Medical Institute investigator. Russell's interests span immunology, microbiology, infectious disease, and molecular and cell biology. His lab studies the interactions between pathogens and the innate immune system. He's interested in understanding how the immune system detects the presence of infectious microbes and the mechanisms by which pathogens evade host immunity and cause disease. So let me now turn it over to you, Russell.
Thank you, Steve. I'm excited to be here to host tonight's event. I think everyone is going to be impressed by the dedication and creativity, and most importantly, by the curiosity that drives the scientists you're going to be hearing from. So as Steve said, the focus this evening is on invisible science. As someone who works on tiny viruses and bacteria that are invisible to the naked eye, I happen to think that this is a great topic, but don't worry, there's no infectious disease talk tonight. I know that we are all sick of that. Instead, we're going to hear from three inspiring scientists, Dave Savage and Gabriel Orebi Gann and Matt Pyle, each of whom work on things that are even smaller than bacteria and viruses. The tiny things that they study aren't always apparent to us in our daily lives, but nevertheless powerfully shape the universe in which we live. In other words, we will learn how small things are actually big. So to kick things off, Dave Savage will first tell us about the microscopic protein machinery that facilitates key biochemical processes inside of cells. Gabriel will then tell us about weakly interacting particles called neutrinos. And to close, Matt will talk to us about dark matter and the powerful detector technologies he's working on to detect it. So buckle up for some fascinating presentations. At the end of each talk, I will try to moderate and have the speakers address all the questions from the audience. So please add questions to the chat. And our first speaker will be Dave Savage. Now, Dave is an associate professor in the Department of Molecular and Cell Biology here at Berkeley. He earned his PhD in 2007 from University of California, San Francisco, and was a postdoctoral fellow at Harvard Medical School. In 2011, Dave came to Berkeley, and his lab now studies very fundamental aspects of cells using the tools of biochemistry, biophysics, and genetics. And he's particularly interested in the mechanisms of carbon dioxide fixation, with the ultimate goal of trying to improve photosynthesis in plants. For his work, Dave has been recognized with a Department of Energy Early Career Award, an NIH New Innovator Award, an Alfred P. Sloan Research Fellowship, and he was recently selected as an investigator of the Howard Hughes Medical Institute. So without further ado, over to you, Dave.
Is that okay?
That looks great. Thanks.
Great. Dave. Fantastic. Yeah, sorry for that cut there.
Yeah.
Thank you, Russell, for the introduction. So today I'd like to tell you all about my lab's work on trying to derive new molecular insights into photosynthetic CO2 fixation or carbon fixation, and how one day we might use those insights to ultimately improve carbon assimilation in biological systems. So I'll remind you that photosynthesis is fundamental to the Earth and human society. And there's an amazing historical connection here at the very small, where on the order of 1 billion years ago, a primordial cyanobacterium, sort of the simplest photosynthetic organisms, were ultimately, in an endosymbiotic event, phagocytosed, brought into another cell, and this primordial evolutionary process ultimately gives rise to the land plants that we are familiar with all around us. This is extremely important because, as you probably are aware, photosynthesis provides all of the necessary ingredients for society in a way. It's the foundation of the food chain. Excuse me. It provides all of the fixed carbon that we use in fossil fuels, and is the major flux for moving CO2 from the atmosphere. In fact, photosynthesis every year fixes more than 10x what human emissions, anthropogenic emissions, actually are. This ancient event, though, actually forever linked plant photosynthesis with this primordial cyanobacterial metabolism. And the interesting aspect of that is it inherited the metabolic pathways, the chemical transformations involved in photosynthetic carbon fixation. And most notably, the most important event in photosynthesis, the CO2 fixation process, where that CO2 molecule becomes an organic molecule, the molecules of life, all the metabolites inside a cell, were linked to one enzyme, ribisco. For those of you who are avid readers, you might be familiar with a recent nonfiction book called The Wizard and the Prophet by Charles Mann. It actually highlights in this book some of the struggles of evolution and its focus on ribisco. The problem of this, though, is that ribisco is really the Achilles heel of photosynthesis. It's the weak link in the process, and it's slow and makes mistakes. For why that is, we don't exactly know. But regardless, it's thought that enhancing CO2 fixation would have a significant impact on photosynthesis in plant photosynthesis. So if we could improve CO2 fixation at this particular step, we could ultimately engineer plants with improved carbon assimilation properties, would be better at CO2 removal from the atmosphere, increased grain yields, and reduced land usage. Many people are looking for various kinds of advancements, and for the last 10 years, my lab has looked actually out towards nature to try to look for interesting features in other biological systems that might be used for these improvements. And we've been focusing on cyanobacteria, which are some of the simplest and fastest growing photosynthetic organisms. Those same cyanobacteria that ultimately gave rise to plants, of course, still exist, They're the sort of the small cells you see here in this micrograph. And cyanobacteria are amazing because they're extremely productive. They are the fastest photosynthetic organisms, and we have wondered, as many others have in the field, why they're so good at what they do. And here on the right, you can see another micrograph, a zoom-in feature. This cell is about a micron in size. And the amazing thing you see in this image of a cell is that we oftentimes think of bacteria as very simple, but cyanobacteria are extremely complex. They have all sorts of interesting structures. And what we and others have shown is that these structures, and here I want to highlight this particular one right here, blown up in a very elaborate, almost geometric looking shape. You'll see what this is in a moment, is part of an elaborate mechanism called the carbon dioxide concentrating mechanism that allows cyanobacteria to be so good at fixing carbon. And the main player in this is something we call the carboxysome, which is a sort of for a cell, it's a massive structure. This is only 100 nanometers in size, so it's actually quite tiny. But this structure, the carboxysome, allows cyanobacteria to be very good at improving carbon fixation. And over a number of years, we, along with others, have shown that how this system works, the carbon concentrating mechanism, is that it uses this carboxysome. Here's actually a 1 million scale model, 3D printed out. You can get a sort of a sense of the amazing regularity of this biological thing that almost looks in many ways like a virus. And we and others have shown that this system works because you have a local concentration of CO2 around rubisco in this structure, and basically force feed the carbon fixation process, along with the help of some other factors like pumps inside the cell. If any of you know about internal combustion engines, you could basically think of this really as a turbocharger on metabolism that's sort of forcing the reactants in as fast as possible. So we have been very interested in this for biochemical reasons, but also for applied reasons, because we want to know how to build them in new cells, not just cyanobacteria. Maybe we could port these advances into other organisms. And when we started our work, turns out we knew biochemically a fair amount about the carboxysome, but we didn't really know what the genetics parts list, all of the other genes that are involved in this process. So we set out to hunt for them. And I won't get too deep into the details, but we did experiments where we took millions of different mutant cells and grew them under a variety of different conditions, say high CO2, where they may not need this special system, and low CO2, where they really need it, which allowed us to genetically identify, these purple dots shown here at the bottom, all of the genes where we could systematically map out everything that was essential for carbon fixation. It turned out it was only about 20 genes, which to molecular biologists like myself is sort of somewhat manageable. And using this information, we were actually first figured out how to be able to build a structure like this genetically by taking these genes and moving them from the native organism into another organism. Thus, we could build carboxysomes, these sites of carbon fixation de novo. Here's a picture of one of those shown here. And more so, this is just the initial step, but as I said, this special structure, this carboxysome, is the site of carbon fixation, but it turns out there's lots of other things inside the cell that also matter. So using that mapping of all the genes, we developed a more elaborate system in which we took all 20 of the genes that were necessary to improve carbon assimilation, and we engineered them in from our carbon fixing organism into E. coli. And E. coli is interesting because it's sort of the standard lab model organism in a typical biological laboratory. But of course, it doesn't naturally fix carbon. It actually normally just grows in our gut. But when we took these 20 genes, engineered them into E. coli, we actually were able to engineer a strain that could literally grow by fixing CO2 out of the air, which is a very interesting proof of concept of how we could get this system to be moved into other organisms. And here's some of that data here showing that it grows in air. What are we doing with this? Well, with this proof of concept in hand and some of the knowledge that we've gained, we're very interested now in taking perhaps the molecules, but certainly the principles, and starting to direct these engineering attempts actually into plants. So we're collaborating with a number of groups at the IGI, where I'm based, the Innovative Genomics Institute, as well as those in plant and microbial biology, to engineer these pathways and inspirations around these pathways into plants, with the ultimate goal of improving carbon assimilation in plants, and addressing some of these challenges in agriculture and climate change. So great, be happy to end there, and I will stop my sharing.
Thanks, Dave. That was terrific. So now we're going to take questions from the audience in the chat. So if you have questions that you'd like Dave to answer, you can put them in the chat. To kick things off, maybe I'll just ask a question. This seems like a very ambitious goal. I'm wondering how feasible it is. What is the chance that we'll actually be able to improve the efficiency of photosynthesis 50% in plants?
Yeah, it's an awesome question, and I think it shows actually the overall strength of Berkeley in answering that, because it turns out on paper there are a number of possible things that one could do to improve photosynthesis in plants. This is sort of something that plant biologists and biochemists have been talking about for some time. There's about 5 or so different strategies that people have mapped out, and perhaps one of the interesting pieces of data we already have is from Chris Niogi here in plant and microbial biology, who's actually shown upstream of this in the so-called light reactions, the reactions where plants are grabbing the photons from the sun to begin with, that other advances from his lab has already, for example, in a recent science paper, shown that you can improve photosynthesis 30%. And so Chris and I are collaborating along with others to take some of our advances, his advances, and start stacking some of these things together. And so the answer is it's actually already possible. And I would also acknowledge the fact that as part of the UC system, we also have access to the extension service. That is the extension with farmers in terms of the California agricultural system. And so as part of this collaboration, we're working with extension people to test these plants in a field as well. So it dramatically accelerates how these things might get into farmers' hands. Great,
thanks, Dave. So we have a couple questions from the chat. Andrea is wondering, I think, a bit about the possible downsides of such a
Yeah, this is a great question. Yeah, it's a very interesting. Yeah, I can handle it. I'll read it out so for folks. Is this just going to create this new toxic version of E. coli that will take everything? It's a great question. So first off, it really is a proof of concept. We needed a sort of a genetic system that we could test where these genes might go. Obviously, what we ultimately don't want is for E. coli to take over. And in fact, as a proof of concept, although it turns out the carbon was coming from the atmosphere, none of the energy that the cell of those E. coli cells was coming from CO2. There's no energy really that a biological system can harvest in CO2. So the energy comes from other input sources. We feed it other sugars, it turns out. So it's impossible for the strain to grow. It has no way of getting energy. But yes, the ultimate hope is really the principles would be ultimately moved into plants.
Okay, I'm relieved to hear that. Dolores is wondering if you could explain a little bit more about this rubisco. Like, what is that exactly? Yeah.
Rubisco, it's a long, interesting story. Rubisco is the central enzyme in the carbon fixation process in a plant. So when a plant is pulling CO2 out of the atmosphere in your garden, on the street, the trees, whatever it might be, that CO2 has to be processed by the cell, and rubisco, which stands for ribulose bisphosphate carboxylase oxygenase, it's the substrates that it uses, is the enzyme that grabs the CO2 and it literally turns this gas into a sugar. So it's a very complicated chemical step, and that represents the fundamental chemical transformation of CO2 into biological systems. And essentially 99% or more of all carbon in biology that we eat, that everything, it all is passed through rubisco, ultimately by plants and by this single enzyme. So it is the most common protein on Earth. A plant leaf tends to be 30 to 50% of rubisco protein by weight. It's mostly rubisco. And it is the most common. There's been recent studies that suggest by mass there's more rubisco on the planet than humans, actually. So rubisco, as an enzyme, is killed and harmed by heat very dramatically. So it would be relatively fragile And so it basically denatures the protein in the same way that cooking an egg white, cooking an egg turns the protein in an egg white. It's the same kind of idea.
I have one other question for you, Dave, if you don't mind. If you describe this photosynthetic process as kind of inefficient, Rubisco doesn't work very well. I mean, if that's the case, why hasn't natural evolution really been able to improve on? I mean, what makes you think you're going to be able to improve on something that evolution hasn't really been able to do so far?
Yeah, it's a fascinating question because it does seem like, so there's a biochemical, a lot of interesting biochemical subtleties to this, but it's a very valid question. Why isn't Rubisco better? Because one might imagine it has more selective pressure on it than any other system in biology, and that's probably true. And that's why for our situation, we didn't try to improve Rubisco directly. We're looking at how nature improves Rubisco. So you don't improve, you can't improve Rubisco, you improve the environment around Rubisco. And when you look in natural plant systems, it turns out that actually some plants do have a system like what I've described, but they do so in slightly different ways. And those have convergently evolved over the last few million years, multiple different times in multiple different plants. And it turns out, unfortunately, it's just most of the crop plants that we grow that don't have this highly efficient system. And so that's where when I say components or principles, what we're really thinking is also looking at how plants naturally do this as well, to combine some of our advances more at the molecular level with some of what is known from the developmental side in plants to combine them.
Okay, great. Thanks, Dave. Looks like, there might be a few more questions in the chat. Maybe you can address those as we go along.
I'll go ahead and answer.
But I think for now we'll move on to our next speaker. So thanks, Dave. We'll move down the size scale a little bit now into even smaller things, subatomic particles. And our next speaker is going to be Gabriel Orebi Gann. She is an associate professor of physics, an experimental particle physicist with an interest in weakly interacting particles. So her research focuses on neutrinos and dark matter, which is about as weakly interacting as you can get. She did her undergraduate work at Cambridge University and then received her Doctor of Philosophy degree from Oxford University. Her primary experimental involvement is with the SNO neutrino experiment, which I think she's going to talk about. This uses a neutrino detector, which is located 2 kilometers underground in a nickel mine in Northern Ontario, Canada. She's been recognized with a DOE Early Career Award and support from sponsors in both fundamental and applied science. So without further ado, Gabriel, please tell us about your research.
Thank you very much. Can you see the slides okay?
Beautiful, yes.
Perfect. So it's a great pleasure to be here to talk to you about unlocking the secrets of the universe with neutrinos. So we know that the sun is powered by nuclear fusion. In the intense heat and pressure in the core of the sun, hydrogen gets fused together and it creates helium. It produces a lot of energy, and that's the heat and the light that we see. And it also produces neutrinos. And the original motivation for detecting the neutrinos that are produced in the sun was actually to help us understand the fusion processes that power the sun. An important thing to note is that all of the neutrinos produced in the sun are produced in what we call the electron state. There are actually 3 different flavors of neutrino, one to partner each of the charged leptons, the electron, muon, and tau. And everything produced in the sun is an electron neutrino. So the first experiments that sought to detect solar neutrinos found something of a quandary. So this figure shows in black the theoretical predictions for the solar neutrino flux, and in blue, the experimental results. The scale on the y-axis is just the fraction of the prediction. You see that the experimental results saw a deficit of a factor of 2 to 3. So where did all the neutrinos go? Do we not understand the sun? Do we not understand the fusion processes, or do we not understand the neutrino? And the experiment that allowed us to understand this question is called the Sudbury Neutrino Observatory, or SNO. As mentioned, SNO was in an active nickel mine in Sudbury in Northern Ontario, 2 kilometers underground, in order to filter out the noise of cosmogenic activity. And SNO consisted of a 12-meter diameter acrylic ball, an acrylic vessel, filled with water, but a special kind of water called heavy water that has an extra neutron in the nucleus. And that extra neutron is really important because it opens up a number of different interactions of the neutrinos from the sun that allow you to differentiate the different neutrino flavors. So SNO was able to measure the pure electron neutrino flux and the total flux of neutrinos independently of flavor. And SNO's results actually earned the director, Art McDonald, the Nobel Prize a few years ago. So I'd like to take a moment to explain why this is difficult. This figure is not to scale. This shows the sun and the Earth, and that's the SNO detector approximately on the surface of the Earth on this scale. It's only 2 kilometers underground. And during the daytime, SNO is observing neutrinos that were produced from the sun, so they're just traveling through the vacuum of space. And then at nighttime, you're putting the entire mass of the Earth in the path of those neutrinos, and you're still looking to detect them. And what's amazing is SNO observes just as many neutrinos from the sun during the nighttime as during the daytime. So that just gives you a hint of quite how weakly interacting these particles are. You can place the entire Earth in their path and they don't even see it. They just go straight through. So SNO's results were definitive. SNO measured the pure electron flavor flux, as I mentioned, and it agreed very nicely. You see it at the bottom, agreed very nicely with other experimental results. But what was really the clincher was SNO's measurement of the total flux, flavor blind, so all of the neutrinos coming from the sun. And that agreed beautifully with the theoretical predictions. So what that told us is, reassuringly, we do understand the sun. We understand the nuclear fusion processes that allow us to survive. But what's happening is that neutrinos produced in the electron flavor, between their production in the core of the sun and their detection on the Earth, they're changing flavor. They're changing state. This is the phenomenon known as neutrino oscillation. And it demonstrates, for example, that neutrinos actually have to have non-zero mass, which pretty much breaks the standard model as it was at the time understood. So that's the kind of result we like to get. We like to break things. So what is still interesting to do with neutrinos? Well, I would say there are 2 areas of interest. One is that because neutrinos interact so weakly, they can actually carry information from regions that we otherwise wouldn't be able to probe. So that includes the core of the sun. There's a lot of interesting physics still to be done there. They can tell us about the explosion mechanisms of distant supernova, and we can even probe inside the Earth. The other area is that neutrinos are a fascinating particle in their own right, and they're actually unique in the standard model of particle physics in having the potential to be their own antiparticle. And that's what we call a Majorana particle. And I use this Escher picture. I think it's nicely representative of this idea, because in this picture, the left-going birds are formed by the negative space of the right-going birds, which is a little bit like what we mean when we talk about the neutrino being its own antiparticle. And if this is the case, this could have very significant ramifications because it actually could explain the origin of our matter-dominated universe. So that's quite significant. Let me explain what I mean by that. So the universe is dominated by matter. If we look around ourselves, we see that the chair you're sitting on is matter. We're all made of matter. There is much more matter than antimatter in the universe. Now, a question is, do matter and antimatter behave in the same way? We actually, many of the interactions, most of the interactions that we're aware of, preserve the difference between matter and antimatter. They'll make matter and antimatter in equal amounts. And if the Big Bang behaved that way, then we would expect the universe to be balanced. equal amounts of matter and antimatter. And if that's the case, well, we know what happens when matter and antimatter come together, they annihilate. So there's a good chance that the universe would not have formed in the way it did. So where did this asymmetry come from? And if neutrinos are their own antiparticle, it actually it allows the introduction of the asymmetry that could lead to the universe having formed the way it did. So the question of the nature of the neutrino, whether they are their own antiparticle or not, has major implications. And it's a really exciting moment in particle physics because no matter the answer, there's something we don't understand. If neutrinos are their own antiparticles, then there's all sorts of new physics to be explored. This means that Majorana particles exist. Are neutrinos the only ones? Might there be something else out there? If neutrinos are not their own antiparticle, then we don't understand where the matter-antimatter asymmetry comes from. And we actually don't understand the difference between the neutrino and the antineutrino. What is it that differentiates the two? So no matter what, there's new physics here and it's very exciting. And what we're doing locally at Berkeley is developing novel technology that will allow us to better understand neutrino behavior and neutrino properties. This image is something called the Eos detector. Eos is the Titan goddess of dawn. And this detector has been designed to demonstrate a novel technology called hybrid neutrino detection, where we use both Cherenkov and scintillation light simultaneously. So this detector is a roughly 4-ton volume inner acrylic vessel, which will house a scintillating liquid. And it's surrounded by novel photon detectors, which are sensitive to single photon levels of light. We're building this on the Berkeley campus right now. And this is a demonstrator for a future detector called Thea, which would be much larger. Thea is the Titan goddess of light, so named because we plan to use different mechanisms of light generation to understand neutrino interactions. And this detector could be up to 60 meters across. That's a 100 kiloton detector. And it would have a very broad range of physics capabilities. So there is an international collaboration of physicists pursuing this project, but it's led here at Berkeley. And we're very excited at the possibility to be able to pursue it. So I will wrap up here just by saying that neutrinos are fascinating particles. They offer us the potential to probe the universe around us in many different ways, while also offering us insights into how the universe may have formed. There's also a societal impact because they may have uses in nuclear non-proliferation. And that's something that we're also exploring in my group. So thank you very much. And I'm happy to take any questions.
Great. Thanks, Gabriel. Fascinating stuff. Our first question, I guess, is from a 5-year-old who's listening. So that's cool. And the question is, what is a neutrino?
That is a great question.
Physics doesn't really have an answer for this yet. But anyway, what's your answer?
Well, neutrinos are fundamental particles, which means, as far as we know, they can't be broken down into anything smaller. Neutrinos tend to be byproducts of reactions. So they're produced in nuclear fusion in the sun. They're also produced in nuclear fission in nuclear reactors. And actually, so I mentioned non-proliferation. That's one way that we could use them for societal impact. You can actually use neutrinos to monitor nuclear activity. So there are tiny fundamental particles that are produced in many places around us. Actually, here's an interesting fact. If you hold up your finger, there's 1 billion neutrinos passing through your thumbnail every second, just from the sun, let alone all of the other sources. So they're ubiquitous.
A little terrifying, maybe, to think of that. But anyway.
But they're not interacting.
Yeah. So this idea that neutrinos might be their own antiparticles sounds very exciting, but how are we going to be able to tell if this is the case? So
there are some processes that will only happen if the neutrino is its own antiparticle. So you may be familiar with the process of beta decay, where a neutron will decay by emitting an electron, and that process also emits a neutrino. Actually, it's an antineutrino. There's a process called double beta decay, which can happen in just a few isotopes in nature, where essentially 2 neutrons decay simultaneously and release 2 electrons and 2 neutrinos. And that process has been observed. If the neutrino is its own antiparticle, it is possible for the 2 neutrinos that would be emitted in that process to actually annihilate each other inside the nucleus. And then the only thing that's emitted is the electron pair. And there's a particular signature for that event. If we see that signature, we know that neutrinos must be their own antiparticle because the neutrinos were not in that event. They must have annihilated each other.
Ava has a question, which I guess sort of follows up from that, asking, what do you mean when neutrinos can be their own antiparticle? Won't they just annihilate themselves? That's what you're saying.
So,
but then I guess maybe the question is, why do they exist at all if they're...
So it's not that a single particle could somehow annihilate itself, but if you shot a beam of neutrinos against a beam of antineutrinos, which if we're saying they're the same thing would be the same, there's some probability that if 2 interacted, they could annihilate. But the probability is not, the probability goes with the inverse of the mass of the particle. And the mass of the neutrino, we actually don't know the mass of the neutrino, but we know that it's many orders of magnitude smaller than any other known particle. So there is a probability that they would annihilate, but it's really, really tiny.
So essentially the reason why they're not just all annihilating each other is because they don't interact very well with anything? Is that kind of the idea?
They don't interact very well with anything, and then even the probability of interacting with each other is even that much smaller.
All right. I was interested in this Thea detector that you talked about. Where is, I mean, this looks rather large, size of the Statue of Liberty. So where is that going to be built?
So ideally, it would be built in South Dakota. There is a mine in South Dakota, a gold mine, which has been turned into a scientific laboratory. It's called the Sanford Underground Research Facility. And there's actually several experiments being constructed there right now to view a neutrino beam that's being directed from Fermilab to that laboratory. And the ideal location for Thea would be there as well, because then we could do physics with that beam of neutrinos, as well as the low energy physics, like looking at neutrinos from the sun. And we'd have a maximally broad program in that detector.
It's too bad you have to go to Dakotas for that. Wouldn't it be better to build it somewhere nice like
Hawaii?
Hawaii, yeah.
I'll take it.
Okay. Okay, well, unless there's any other questions, we will, I guess, thank Gabriel for a fantastic presentation. And if there are more questions that pop up, maybe you can address them in the chat. But now we'll move on to our final speaker for tonight, Matt Pyle. Matt is an assistant professor in the Department of Physics. He did his undergraduate work at the University of Notre Dame and received his PhD at Stanford University. His research interests include astrophysics and nuclear physics, and pursuing questions like, could dark matter be composed of particles with mass less than of a proton? His goal is to develop these new detector technologies and employ them to find answers to these kinds of questions. So currently his group is focused on optimizing the design of massive low temperature calorimeters for the SPICE, HERALD, and Super CDMS low mass dark matter experiments. And I assume he's going to tell us something about that. So take it away, Matt.
Hi. So let's talk about dark matter. So before we talk about that, let's just go back to the 1600s, and Newton told us something exciting and told us about F equals ma and the law of gravitation. And what we saw is that we could explain how all the planets revolve around the sun and how gravity pulls the planets and into the sun, and they sort of spiral around and they orbit. And we actually, since the 1600s, we've been able to actually measure the period of all of these planets, or equivalently the speed of these planets as they revolve around the sun. And what we see is that the farther out you go, the slower the planets travel. And this exactly matches what we would expect from the law of gravitation, F equals ma. So in the 1970s, Vera Rubin did exactly the same thing, but rather than with our solar system, she did it with galaxies. So here is a galaxy, and we can think of the center of the galaxy where all the stars are as sort of like the equivalent of the sun. That's what's pulling all the stars in. And then we can look at how fast the outermost stars are rotating around that galaxy. And what we would expect is this shown in black. So in the core, the further out you get, actually, the stars will speed up because the core is getting bigger, sort of like the star is more massive. But then once we're out of the area where all the stars are, we should see exactly what we saw in the solar system, where the farther out you are, the slower the stars go. And what Vera saw was instead these very far out stars kept on traveling really, really fast. And what this means is that there's some mass that we can't see, and I've shown that here in red, but there's just a bunch of particles that we don't see that are pulling these outermost stars in towards the center and keeping them traveling really, really quickly. So the stuff we can't see, we call dark matter. And it's not only Vera's work, but there's an enormous amount of observational evidence that dark matter exists. So I'm not going to explain these plots. We can talk about them later if you like, but Big Bang nucleosynthesis, we've measured a bunch of like how many atoms were made when atoms were originally condensing out of neutrons and protons in the early universe. We can look at the cosmic microwave background, and it exactly matches how much dark matter we would expect. And likewise, we can look at clusters, and particularly the bullet cluster, where two clusters colliding together, which shows that we definitely have dark matter. And so why is this so important? Well, first and foremost, it's a large fraction of the universe by energy. So the stuff we're made out of, which is neutrons and protons, is something like 5% of the universe. By energy, it is 5 times that. So that's what's shown here in blue. The rest of the purple is dark energy, another dark thing we don't understand. So the other reason, just beyond the fact that it's such a large fraction of the universe, is that it is the one particle that we know of outside of the standard model. And because we know it exists, if we can play with it, if we can interact with it, if we can understand what its mass is, how it interacts with us, then hopefully we can understand more about the laws of physics at very high energies that we haven't been able to probe, and that we know our current laws of physics are somehow incomplete. So the hope is that dark matter, if we can play with it, will become this Rosetta Stone for how the laws of physics really work at very high energy scales. So let's now talk about what we mean when we call it dark. And to do that, we can think about 2 processes. Like when when you listen to a radio, there's an antenna somewhere which is shooting electrons back and forth across the antenna. And this jiggling of electrons, they're charged electrically. That jiggling produces radio waves, which you're going to see, which you're going to hear on your radio. Likewise, when you look at a mirror, what we have light coming in, which is a wave, and it interacts with the electrons in this metal, and those electrons jiggle, and they create a wave, a reflective wave, and another wave that cancels out the internal wave, the wave coming in, and that's what a reflection is. So when things reflect, and when photons are created, right, what that means is it happens because electrons are charged. So when we say something's dark, we're saying something deep. We're saying it's not charged electrically. And equivalently, we're saying it's not charged in any of the current forces we understand. So it's not charged into the weak force, it's not charged into the strong force, which holds nuclei together. Dark matter doesn't have any of those forces. So that means it really is invisible. So to all the forces that we currently know. But we still hope that it's going to scatter off of us every once in a while. And so I build detectors to see these very rare scatters of dark matter hitting, like if we're here, a crystal, and scattering off and leaving a little small amount of energy. So one of the things we don't know about dark matter is the mass of that particle. All we know is the mass density in our universe. And my lab specifically looks for dark matter in the mass range from something like 10 proton scale of mass to something down to basically the neutrino scale that Gabriel was talking about, or something like, I don't know, 10 to the minus eighth of the electron mass. And so what is the challenge of looking for these, of this mass scale of dark matter? And it's actually, again, a very simple Newtonian mechanics problem. If we have a light mass dark matter particle scatter off of a big nucleus, the energy deposition is very, very small. The energy deposition for something like dark matter of 1/10 the size of a proton is something like 10 to the minus 19 joules. And what is 10 to the minus 19 joules? That's something like the energy needed to lift a paperclip 10 to the minus 17 meters. So it's a very small amount of energy we're trying to see. And another way to think of this is sort of like the vibrational energy of, like, 10 or so atoms. So how do we measure that? Well, we're going to make a calorimeter, but it's going to have to be a very good calorimeter because the energy that we're depositing is so tiny. So we're going to want to have a really small heat capacity. But in some very reasonable analogy, this is sort of like having a pot of water, and you put energy in and you measure the temperature difference. So we just want to have a much more sensitive version than that. So how do we get that very sensitive thermometer? And the first step is we want to go very, very near absolute zero in temperature. And you can sort of see that qualitatively why here. Temperature is just random vibrational energy. So all of the atoms in the table that you're sitting at in your chair, they're all wiggling around. And the energy they have is proportional to the temperature of the system. And so when we're looking for this little small amount of energy given to this atom here in red, it's going to get swamped out by all these other random vibrations. So we want to get rid of that by cooling it so we can see this red part. And so the first thing we do is we cool to near absolute zero, or 6 milK above absolute zero. Then we build the world's most sensitive calorimeter, and here we use actually a bunch of superconducting sensors. And you see on the surface of this crystal, we've used nanotechnology, the same that you would use, for example, if you're making, I don't know, a transistor or like a computer chip, to fabricate little superconducting microphones on the surface of this detector. And that calorimeter is this very small 1 micron by 25 micron by 40 nanometers thick tungsten line here at the center of each of these very small microphones. And that's the thing that we heat up and we measure its temperature of. And then the final thing is, if we want to make ourselves sensitive to these very, very tiny energy depositions, we have to get rid of everything else. So in fact, here is a cell phone, I guess, from like an undergrad running around campus. And this is from probably 4 years ago, where we could actually see that cell phone in red. We could see a packet of data being sent, and some 10 to the minus 16th of that energy that's given off by that cell phone is actually somehow getting into our detector and heating those calorimeters up. And that's actually shown here in red. So you could pulse shape reject it, but the answer is we just want to filter it out. Likewise, vibration from our refrigerator could also heat these things up. And so you can see that here we actually hang these crystals from little wire bonds to isolate the system from vibrations. And then finally, step 3 is we go underground. And this helps get away from all of these cosmic microwave backgrounds that come from outer space that could hit our detector and heat it up. So those are the 3 things we do to make our detectors super sensitive. And so I think here's a summary. Understanding dark matter is important for these 2 reasons. It's a large fraction of the energy content of the universe. And it allows us, hopefully, if we discover it and play with it, to have insights into the laws of physics at very high energy. Its mass is completely unknown. So depending on the mass of the dark matter, your experiment's going to look different. But for our experiment, the dominant problem and the why people haven't been able to like study and look for dark matter in this area previously is because they've never had detectors that were sensitive enough to see these depositions. So that's it.
Great, thanks, Matt. That's terrific. If there's any questions for Matt, you can put them in the chat and I will pose them. But to get things started off, I'm wondering, in this whole attempt that you're describing to really build these detectors to detect this dark matter, have you stumbled across anything unexpected? Things haven't gone according to plan, I assume, but maybe you can.
Things never go to plan in physics. I mean, physics is failure, right? I think that's actually science is failure. You always fail many times before you succeed. But I think there's actually probably the most interesting thing that's happened is what we mean. I talked about this idea of as you cool down, we expect random vibrations and all this randomness to disappear and everything to be very clean and simple. And this is needed not only for us when we're studying dark matter and trying to see small energy depositions, but this is also really important for things like quantum computing, where we're trying to keep a quantum state coherent and to use it to compute things. And so one of the things that we found in our detectors is we see random microfractures, so random sporadic little dissipations of energy pulses that hit, and we've correlated this to some quantum computing groups that they also see something similar in their background, and so this could be an ultimate limit of quantum computers. So in that sense, our work is not only fundamental, but it's also very applied in that if we can figure out what these microfractures are and how to get rid of them, and we actually have a very good idea of where they're coming from now, it could also help the creation of a quantum computer.
I love when things lead in unexpected directions. That's great.
Yeah.
There's a question from Rosette who's asking if there's any relationship between dark matter and black holes. They kind of sound similar. Is there anything?
Yeah, there is. I mean, both interact gravitationally, so that's the first one. But actually, secondly, there are theories that maybe dark matter is actually just a bunch of black holes that are flying around at various mass scales. So one possibility for dark matter could in fact be that it is black holes. So that's another interesting combination.
And lastly, I have a question about how dark matter really is. I mean, if it's truly dark, how are you actually going to detect it?
Yeah. So that's actually, because if it's, we talked about what we mean when we say dark, and mean it's not charged. And so this is actually one of those things where there is a little bit of a hope here in play. I mean, we hope to see dark matter because it would tell us so much about the universe. But at the same time, there's a reason why we would expect to see dark matter, and that is because dark matter was somehow created. And for it to have been created, as the universe at the Big Bang or after the Big Bang at some point, that means it had to have interacted somehow. And so we believe that at some energy scale or via some mechanism, it interacts with us because it was created at the same time we were. At some level. So the hope is that that process that created dark matter in the first place is also going to allow us to see it now.
And maybe that sort of gets at a question in the chat from Ava, who's asking, is dark matter rare or hard to find? She says most of the universe is made of dark matter. Why? I guess the question is kind of like, why is it so hard to see?
I know. No,
so much of it.
So again, so it is certainly not rare. It is 25% of the universe. It's hard to find because it's truly dark. It doesn't interact with us via any of the known forces. So just like Gabriel was talking about neutrinos can go through the entire Earth without interacting. So in some sense, dark matter is the same idea, that it interacts so rarely, it just goes through us all the time. And we're looking for that once in a blue moon interaction where it doesn't go through us, but it's an incredibly rare process.
Okay, fascinating. Thanks again, Matt, for that great presentation, and to all the presenters. I think we've learned a lot about invisible science, things that maybe we don't think about in our daily lives, but are, happening all around us all the time. So to close, I'm going to hand the floor back over to Steve Kahn, the Dean of the Division of Mathematical and Physical Sciences, to wrap up. Steve?
So thank you, Russell, and thank you, of course, to David, Gabriel, and Matt for sharing with us the things you try to see and the tools you use to find them in this invisible universe we've been hearing about. I really enjoyed tonight's discussion and finding the parallels between so many different kinds of science that occur on the Berkeley campus, both in the life sciences and biosciences and in physics. As I said, this is the last event of the fall series, but we are in the midst of planning for the new year. We will no doubt showcase another round of excellent faculty and students as our Basic Science Lights the Way series continues in 2023. And of course, please reach out to us if you'd like to learn more about anything we've covered. We'll be sharing resources with you by email about today's topic and speakers. And a video of today's session will be available at our Basic Science Lights the Way website, which is basically basicscience.berkeley.edu. That posting will be approximately a week from now. You can always return to that website to watch any of the episodes that we've previously had that you were unable to attend. They're all available there. So with Thanksgiving around the corner, I thought I'd take this opportunity to thank everybody for showing up at these events. We really appreciate your attention and your interest, and the appreciation of the research and discoveries that we've generated here at Berkeley. We couldn't do it without you and with your support. So until next time, stay curious, fiat lux, and go Bears. Thanks very much.